language. So we looked at respiratory function, we looked at hand function, we looked at all of their functions of daily living to make sure that nothing else above was changing. But we also did electrical stimulation down here and measured up here, so that sensory, we stimulate the sensory nerves there and recording up here. We didn't see any changes there. And we also did magnetic stimulation up here to stimulate motor pathways, which should affect the muscles, and we didn't see any of those changes. So nothing dramatic happened. Next slide, please. One of the transplant patients did find an increase in sensation below the level of the injury. So, as most of you will know, in the ASIA score is defined usually by the physiotherapist in a very standardised way, and so below that ASIA level, the neurologist, and we had a consulting physician who wasn't blind and who oversaw the health of all the patients throughout, and the patient all noticed that there was a change in sensation below the level of injury, and as most of you know, there's this often a zone of partial preservation. What we think is that the cells in this person may have affected the zone of partial preservation. But that's 1 out of 3 of the transplanted cells. It's hardly a statistical number, so we have to say these are very preliminary. Some of the preliminary nature is because we've only got 3, even though we wouldn't expect changes to occur, they could occur because it's a rare event in a potentially large population. Next slide, please. So we have a small sample size. One of the preliminary aspects of this too is, and this might be unresolvable in human trials, we don't even know if the cells were there. We don't, because you inject the cells in, and if you do that in the rat, they stay in there, and you come back later, you can chop up the spinal cord and you can see the cells, but of course we can't see that with humans. So that will be an unknown for all cell therapies in humans. Whether or not the cells got in there is a major issue, I think. Well, not knowing that they're in there is a major issue. So the surgical procedure is safe. The olfactory biopsies and transplantation seems to be safe. And we'd say at a preliminary point here, the cells might improve function, but that's not yet proven. This is to remind us that these things happen when you have a large group of collaborating people, clinicians, scientists, patients, clinicians of all different kinds. I was the scientific director with overall management of the scientific side, and Tim Garrity, who's the head of the spinal cord injury unit in Brisbane, was the medical director of the trial with overall responsibility for the patients. And we had a big planning team. And we started thinking about this in 2001 and getting the ethical approvals and getting the money. We transplanted the first patients in 2002. And we did this 2 by 2. We had, well, 1 by 1 over several years. Francois Ferrand, who was the cell biologist who developed the culture technique, he went back to get a job in France in the middle of it. That's my 2 minutes left. 2 minutes. Gosh. Anyway, big trial, big numbers. I better get on to the Geron trial in 2 minutes. So the Geron trial opened. They spent 10 years getting animal evidence and getting Food and Drug Administration approval in the United States. And in 2010, in October, they started the trial in humans. So they'd taken them, at that time, Tom O'Connor, who was head of CEO of Geron, said they'd spent $40 million and they had a stack of regulatory papers about this high to get to this point. Next slide. What they did was to take embryonic stem cells, and then they would make from them these oligodendrocyte precursors. Now, the oligodendrocytes are the cells that insulate the nerve fibres in the spinal cord. And they did animal studies, only some of which have been published. Next slide, please. They did animal studies that showed that here's the injury side, and they injected their cells, and basically those brown cells will be the oligodendrocytes that have developed from the cells that they put in, and it alleviated spinal cord injury in the animals. And then, not published with all the work they did for the FDA, was to prove that there were no cancers or tumours or all the safety aspects of putting these cells into the animals. So the trial started in October 2010. Next slide, please. Remember, our trial was what we call autologous. You take cells out of somebody, you grow them, and you put them back into themselves. They said, and this is really probably the commercial reality, if you're going to do this in a commercial way, you want the cell in the bottle. Somebody has an injury, you've got a cell, and you take it off the shelf and you put it in. So they developed this, and this is based on the kind of drug model. You have a cell or a drug, and each of those products like that have been approved, they've been safety checked, they've had all of these. So each individual product has had all of this testing to make sure that this batch number is suitable for human use. Next slide. I won't go into all the details, but basically their patient selection was the same as ours, except that they were choosing patients within 7 or 14 days of injury. So they were putting these cells, they were injecting them into the spinal cord, but now in that very early stage. And again, they were doing a safety trial, and they don't have any controls, they're just putting the cells in and then following the progress of those people. Ostensibly, so they had non-randomised, not controlled, not blinded, 7 patients they set out to do, and they were going to follow them for 15 years because of the potential for tumour formation and other safety issues. Next slide, please. So, again, they had all the same surgical risks as we did in our trial, but they had certain risks that need to be thought about for embryonic stem cell research. First of all, and this particular trial, it was an early trial, so it was happening in that early, very early stage in the acute injury, so I've discussed the issues there. But because they're taking cells from another organism, another human, or at least human-derived, they need to suppress the immune system, otherwise the person will reject those cells as a foreign transplant. So those are special risks, and there's certainly risks that are important for people with spinal cord injury. And then the other issue is, and they've proven that these cells didn't make these tumours, teratomas they're called, in animals, but of course that's always a risk with these cells when you take them into humans. So those are extra risks of embryonic stem cell transplantation. So in October, a year later, they reported that they've transplanted 4 patients. Next slide, please. And that there were no surgical complications after those 4 patients. Next slide. And then a month later, the trial was terminated. Tom O'Kama, who led all of this and pushed all of this, was, for whatever reason, was no longer the CEO.